Thank you very much for having me. I'm Ryan Mitchell. I'm the Director of Molecular Biology here at InnerPlant. I'm a molecular biologist by training. I've spent the past 12 years working at various startup biotechs, first at a company down in San Diego called Synthetic Genomics, where I focused on ag microbial discovery. Then I took a hiatus from ag and then worked in human health in the gut microbiome before transitioning back into ag and ending up at InnerPlant, where I've been for the past 3 years. And so at InnerPlant, we're turning plants into living sensors that we can see from space. And we'll touch on the last component of that a bit later in the talk. So the dirty little secret about ag is that farming still largely operates like it did roughly a century ago. Sure, there's been advances in new tractor technology, new fungicides, and new herbicides hitting the market, but a farmer is still only able to diagnose what they can actually see in their fields. And many times when there is visible symptoms in the field, it's already too late and damage and yield loss has already occurred. And so to adapt, farmers typically preventatively spray chemicals everywhere across all of their fields. And there's roughly a 40% global yield loss annually, which represents around $220 billion in economic losses. And additionally, there's 30% of chemicals that are potentially wasted. And not only is this a huge environmental problem, but it's more than a $200 billion problem as well in nitrogen loss to the environment. And so at InnerPlant, we have the idea that we're able to grow twice as much food with less chemical inputs. But how do we do that? And so constantly at InnerPlant, we're asking ourselves, what if plants could actually tell us what they need? And so here at InnerPlant, we're developing a novel patented seed technology that we embed signaling capabilities within the plant genome, which allows for a triggering of a fluorescent signal within hours of a stress, way before visible symptoms are seen on a plant. And so we tap into the plant's physiology by harnessing early and specific gene activations that happen within hours of a detected stress. We then insert lines of code or lines of DNA into the plant genome, and that allows the plant to produce a new protein when it's stressed and give off a fluorescent signal. And by doing this, we offer novel sets of data to farmers and clear data-driven frameworks to detect, diagnose, and act on stress. So let's dive a little bit into some plant science and specifically around plant stress. So plants are always responding to their environment. And in the case of stress, there's multiple different kinds. We at InnerPlant categorize them by abiotic versus biotic stress. And so abiotic is nutrient deficiency or drought or something like that, whereas biotic stress is a fungal pressure. There's insects eating your plant, nematodes in the soil. And so when a stress occurs in a plant, genes start turning on and off, meaning that genes are transcribed from your DNA and then translated into protein, which then lead to some sort of adaptive response, whether, if we have fungal pressure, some sort of disease resistance, the plant's going to try and fight off of that fungal infection, or potentially modified growth if there's nutrient deficiencies, changes in photosynthetic activity, metabolic adjustments, and so forth. So how do we actually measure these changes in gene expression? RNA sequencing allows us to show that very specific sets of genes are turned on to very specific stresses. So let me give you some context on how to read this matrix. And so across the top are 5 different stresses that we've imposed on plants. So we have drought, pathogen stress, insect pressure, phosphorus deficiency, and potassium deficiency. And then across kind of the y-axes are sets of genes that we've identified that are specifically upregulated or turned on during these stresses. So when we look at the drought, the drought genes, we can see that the genes highlighted in green are significantly and highly enriched and upregulated during drought stress. But it's important to note that they're not turned on during pathogen attack, insect pressure, or various nutrient deficiencies. And that pattern kind of holds across these different stresses. So we have sets of genes that are specific towards pathogen pressure and are downregulated or not affected by other stresses. And so how do we actually do this? So we actually tap into the plant's natural response pathways by combining key genetic regulatory elements with fluorescent reporter genes. And so to dive into a bit of more of plant molecular biology, we have a gene here, highlighted here, and then we have the upstream region of the gene and then also the downstream region of the gene. And we'll dive into what is kind of in each one of those different regions. So upstream of a gene is, we have the potential for enhancer regions, which either attenuate or help translation of our gene of interest. And then we have promoter regions, which are basically the on-off switches of our gene. And then we actually lead to the transcriptional unit, which is actually transcribed from DNA to RNA. So we have a 5' UTR, which contains potentially some regulatory regions, but also helps with stability of and translation of your RNA. And then we have the protein coding sequences, which actually make the protein that has that downstream function that we're interested in. And then we have the 3' UTR, which does a lot to do with like stopping translation, but also can affect protein stability as well. And then downstream of the gene, we have the potential for additional enhancer regions. And so at InnerPlant, we take these regulatory sequences that are upstream of a gene, basically the on-off switches, and we hook those up to a fluorescent reporter. And then so when we introduce these cassettes into the plant and there's a stress, our plants then fluoresce. And so I mentioned that we have this idea that we can see fluorescent soybeans from space. And to do that, you could imagine that we need very fast signal and very bright signal. So some of the things that we do in the lab are random mutagenesis or targeted mutagenesis of our fluorescent reporters to make a brighter signal. And so we do use AI and particularly AlphaFold to understand how the different variants that we're making look on a protein like structural level. And so in this slide, we have a model system in Arabidopsis of an insect sensor that we've built. So this is a Petri dish of our insect sensor. So on the left, we have a treated group, which have been sprayed with a hormone that simulates insect pressure. And then on the right, we have a mock treated group. And then we have a video, hopefully that will start. That's actually a time course of fluorescence. Let's see if it'll start. There we go. And so you can see that, so it's a 48-hour time course. And you can see that the plants on the left, which have actually been sprayed with the hormone, get very bright very quickly, whereas our mock treated plants don't. And so at InnerPlant, you know, we've worked in model systems in Arabidopsis, but we want to move into something that's like economically viable and important for farming practices. And so we're very focused on soybean and specifically soybean fungal pressure. And so one of the things that InnerPlant is doing is making a signal that's actually detectable before we see visible symptoms. And so these two plants highlighted, or the RBG images here, are one is a mock treated plant and one is a plant that has been sprayed with fungal spores. And we can see on the graph to the right that one of these two, we get really high signal within 2 days of inoculation. But it's important to note that there's no real difference in the RBG image at this point. There's no lesions on the leaf that we can differentiate between. But when we look fluorescently, there's a big difference. It's quite obvious to tell that the plants that have been sprayed with the fungal spores, they're actively making our fluorescent reporter. And so this is great. You know, we've been working in the lab. We've got really cool fluorescent proteins that are being made in plants. We've got glowing soybean plants. But who really cares unless we can actually get this into the hands of farmers? And at InnerPlant, we care about the farmers and we want to do everything that we can to help them out. And so how do we actually get this into the hands of the farmers? And so this year we're introducing CropVoice, which is powered by our inner soy fungal pressure soybeans. And what does this actually look like? So for CropVoice, we are deploying a network of what we're calling sentinel plots. So plots that we manage and manage the detection of fungal pressure. And so farmers enroll through their local retailer. And once they're enrolled, their plots are incorporated into our network. And so when there is fungal pressure in the area, a farmer gets an alert on their phone that, hey, there's fungal pressure in the area. Please go speak with your agronomist about next steps. And so just to reiterate kind of how, the technology works is fungal infection occurs in the plant and it activates the plant immune system. And then that DNA triggers the fluorescent protein to be made, and there's an actual fluorescent signal that's given off. And then disease is confirmed by detection and a couple of downstream methods. So one thing I want to introduce is the idea of the disease triangle, which is the idea that you need a susceptible host, a virulent pathogen, and environmental conditions that favor the pathogen to be able to yield disease. And so farmers traditionally have been able to understand two parts of that disease triangle, but they're not able to close it all the way. Farmers are able to understand, what potential pathogens are in their fields with soil testing or spore cams. And then farmers also understand the weather and what kind of environmental conditions are happening in their fields. But without InnerPlant and our inner soy technology, they have no idea what's happening within their plants at any given point. And so we're trying to change that. And so what does being enrolled CropVoice actually look like? So let's dive into the first point. So plants signal infection, right? There's going to be a fluorescent signal. We're monitoring these fields 24/7 autonomously. And how are we actually doing that? So our engineering team has had to come up with a way to develop durable, replaceable hardware that can be deployed in a field and that allows for continuous monitoring for infection. And so what they've come up with are these what we call light bars, are 5-foot kind of lights that are held up on a couple different legs, and then we deploy multiple light bars throughout the field, and then we have multiple fields within our network that are kind of all monitoring everything. And so you can see just a couple example images of the RBG images that you get out of the plant, out of the light bars, as well as the fluorescent images. And in this case, this is a flat of soybean plants that have been sprayed with a fungal spores. And so you get really bright fluorescence. All right. And then, so this is a new product that's hitting the market. We want to be able to basically ground truth, what's actually happening with our plants as well. So we're spinning up a in-house soybean pathology lab. We have local agronomists with decades of experience on staff that are experts in soybean disease and agronomy. We also have some weather data modeling, and then we also have some kind of advanced modeling around disease forecasting and things like that. And so to point out, one of the aspects is our implementation of the pathology lab, which is basically to backstop any of the data and validate any of the data that we see coming off of our light bars. So we're doing a couple different things. We're taking a culture independent approach as well as a culture dependent approach, meaning that in the culture independent approach, we don't actually need to isolate the pathogen on a petri dish. So that relies on some novel molecular assays for species level discrimination. So there's a couple of soybean diseases, specifically frog eye leaf spot and Cercospora leaf blight, which are all caused by species within the genus Cercospora, but we need to be able to identify the causal species. And then we're also deploying a culture dependent method or methodology, which is basically what you would get when you work with like a university extension program. So basically the gold standard for pathology. And so one of the things that we're doing with this data that we're capturing is understanding like, what's actually causing disease in our plants. And we see a world where, this could be data that we would use for model training. One specific thing is, are there different fluorescent patterns that we see that are tied to different specific pathogens? That's something that we're trying to learn, but could be used as good data sets for model training. And then lastly, we've detected signal, we've validated signal. How does this actually get into the hands of the farmers? So farmers receive a notification in a couple of ways. So if fungal pressure is detected in their area, they get an alert on their phone saying, a text message saying, there's fungal pressure in your area. Please go speak with your agronomist about, what your next steps might be. And then we've also integrated into the John Deere Ops Center for the farmers that are connected that way. And so one of the ideas that I want to highlight is the idea of a leading indicator versus a lagging indicator. And with a lagging indicator being something that a farmer learns at the end of season about what actually happened during their whole kind of growing season. And nothing with that is actionable until the next growing season. Whereas at InnerPlant, we're providing like actionable data-driven insights within like the growing season that a farmer could, get an alert and a day later jump in their tractor and start spraying a fungicide. So yeah, we're trying to connect to the farmers fast and with data-driven insights. And so in 2025, we are actively starting right now. It's the starting of planting. So we are deploying 2 commercial networks where we're actively enrolling farmers and growers. So we have one in central Illinois, and then we have another in Northeast Nebraska. And then we have a couple kind of what we're calling experience plots and experience centers where we're holding field days throughout the season. All right. And then the last slide, or the last thing and kind of the sci-fi moment is, can we see soybeans from space? The short answer is yes. And, in the next kind of image that's shown in the video is an actual data point from remote detection of our field here that's highlighted of fluorescently glowing soybeans from a satellite. And so we need to move, be able to scale our technology. And the easiest way to do that is with satellite in terms of covering the most acres. There's other technical challenges, around making plants that are actually bright enough to see from space. But right now, we have been able to see our soybeans from space. So, it's a great step forward, but you can also imagine a world, in the future where all of this is done autonomously. So you have, a swarm of satellites circling the globe that are actively monitoring your plots. And as a signal is detected, they send something to an agent or a signal to an agent saying there's pressure in these fields, and an autonomously driven tractor drives to that field and then has a kind of precision see and spray technology for spraying. And as it drives through the field, it's in real time evaluating plants, looking for fluorescence, and only spraying the plants that have, the stress. So this is a world that we're moving towards, and definitely it's in the future, but, AI is definitely part of this future. And so just a couple other ways of how we're incorporating AI into our system is, we've incorporated AI into all of our code bases, which basically allows us to like rapidly deploy new algorithms and new models, and just basically increases the speed of software development. And then right now we have simple but robust algorithms that we know are going to work for detection of fluorescence. But in the future, we're building these big data sets that we can then build models for around disparate data streams that we hope can converge and perform additional insights, such as all of the various agronomic data streams, our fluorescent plants plus satellite and pathology. So I just want to thank my team and the whole InnerPlant team. It's a multidisciplinary team made up of biologists, engineers, software developers, and sales that actually get this into the hands of farmers. So at InnerPlant, we're fundamentally trying to change the way that we farm by unlocking new data-driven systems built around what crops actually need. Thank you.